So actually, why the SNPs are important? Like why identifying the SNPs or like a variation is important in our uh, why it is important? Because actually the SNPs can be used as a biological markers, means like used to identify to locate the genes which are associated with a particular disease. And whenever you are getting any SNPs which is occurring in any particular region or region of gene or any regulatory region, so they play a more direct role in a disease by affecting the gene function. That's why if you are dealing with that, you can go with the DNA-seq analysis. So what is the workflow of the DNA analysis? That we will see first of all. So the first thing or first step, like you have to collect the raw data. Okay, like whatever the raw data you are getting, so you can either get it from the database, or if you are directly sequencing it from the machine, like you have your own data, so that you can take. So that data, it will be in a FASTQ format. How the FASTQ format will look like, what it contains, that I will explain you. But the raw data, it will look like in a FASTQ format. Then once you are getting that, you have to perform the quality control, because all the data or the bases are detected by the machine, so you should know whether they are having a good quality or they are not having a good quality. Is there any like adapters are present in the data, or maybe any bases with a low Phred quality score or low base quality score? So that you need to be identified, and that you can perform with the tools like FastQC, MultiQC. There are so many tools we are having. The choice of the tools is depend upon yours. Then maybe you are getting the two kinds of output, like your data might be a good quality or a bad quality. So if your data is having a bad quality, you can go process further for the trimming. So for the trimming, you can trim the low quality basis. Maybe if any adapter or primer is present in your data, that you can trim out. Maybe if any sequences with a very like less length, or they are very shorter sequences, you can trim that. Depend upon what, like how the data is having a problem, you can use the tools Cutadapt or a Trimomatic for that, or a FastP for that. So once you're sure, yes, you're like, once you trim the data, what you can do, you can again check the quality to make sure, yes, your data is having a good quality. And once you're sure, yes, your data is having a good quality, then you can process further for the mapping. Okay. So basically in a mapping, you need a reference genome. So you first need to get the reference genome in any one of the database, and then you can align your reads to the reference genome. But actually, whenever we are doing an alignment or a mapping, there are two steps we have to do. First, you need to do the indexing. Indexing means what? Like whatever the reference genome you are having, that you are going to divide into a short sequences or short files, because when you are aligning the reads to the reference genome, like this genome is very large as compared to the read sequence, and that's like very shorter sequences also, or short data also, it is going to take a very long time. So that's why we are converting our reference genome into a small binary files, which will increase the speed of mapping. So like whatever the output files we are getting for this indexing, that is in a binary format, which we are not able to understand. So to those binary files, or you can say to that index genome, then we are aligning our reads. The choice of the tool is up to you. Like there are so many mapping tools we are having. BW we are having, Bowtie we are having. So depend upon whatever the choice of the tools you are having or you wanted to use, you can go with that. So once you are getting the mapping result, so that mapping result you are getting either in a BAM format or in a SAM format. So what is that? That also I will explain you in the next slide. So once you are getting the mapping result, after that, what you have to do is you have to convert that file into a sorted format. Whatever the mapping file you are getting, that you have to convert it into the sorted format. And then from that file, you need to be remove the duplicate reads. So because whenever we are doing the sequencing, we might get some duplicate reads. And if you wanted to identify the variation, because here we are doing a base-wise alignment, right? So we cannot prefer to keep any duplicates in our data. So you can remove the duplicates. So again, for that, we have the SAM tools also. We have a Picard tool also. So depend upon that, you can use. And then you can call the variations using the SAM tools, using the BCF tools, using the GATK, depend upon the tool choice you want. And once you are calling the variations, the output, it will be in a either BCF format or in a VCF format. Okay. So the VCF again, and what is a BCF, that also I will explain you in the next slide. So this is a general workflow for the DNA sequencing. Now,